Hi viewers, this is Karunakar, lecturer in botany. Today we are going to discuss about viruses. What is the definition of virus? The most important topic in this today. To see the virus, these are obligate intracellular parasites which can only be reviewed with the help of electron microscope. They are so small that they can pass through the pores of bacteriological filters. Characteristically, they possess only one type of nucleic acid, either DNA or RNA. The Adolf Meyer, a Dutch scientist, and D.J. Ivanovsky, a Russian scientist, recognized that certain microbes other than bacteria are the causative agents of mosaic disease of tobacco. The basic criteria used to differentiate these agents from other familiar microbial agents was their ability to pass through bacteria through filters developed by Chamberlain in 1884. These agents were thus designated as filterable viruses, that is, poisonous fluids. M.V. Bejering, a Dutch microbiologist, demonstrated in 1898 that viruses differ from other cellular organisms. He discovered that the virus of tobacco mosaic disease could be precipitated from a suspension of alcohol without losing its infectious power, and the fluid was capable of diffusing through agar gel. This led Bejering to believe that the fluid itself was living, and he put forward the concept of contagium vivum fluidum. This idea was considered revolutionary because for the first time it was acknowledged that there occurs a new type of disease causing agent. In the same year, the Froch demonstrated that the foot and mouth disease of cattle is caused by filtrative apparently free of any bacteria. And if you see the particular definition of viruses, the virus constitute a large heterogeneous group of infectious agents that differ from other microorganisms in numerous ways. They are acellular microbes as they lack cytoplasmic membrane to separate them from their surroundings. They are obligate and intellectual parasites and incapable of any activity without a host. They totally depend on compatibility host cells from their replication. Outside the host cells, they are essential non living particles various within the host cells. They exhibit characteristics of living systems. Then, how a virus may be defined. The definition of viruses itself is arbitrary, and in fact, many definitions have been proposed from time to time. And Lowe defined virus as strictly intracellular and potentially pathogenic entities with an infective phase and interface. They also possess only one type of nucleic acid, and second one is multiple in the form of their genetic material. Third one is unable to grow and undergo fission, and fourth one is devoid of a Lippmann system. This definition stresses the non-cellular nature of viruses, their dependence on host cell metabolism. With the advancement in the knowledge of virology, a more satisfactory definition was put forward by Luria and Dornell. This states that viruses are entities whose genomes are elements of nucleic acid that replicate inside the living cells using the cellular synthetic machinery and causing the synthesis of specialized elements that can transfer the viral genome to other cells. This definition conveys two essential qualities of viruses: possession of a specific genetic material for replication inside the host cell, and the second one is possession of an extracellular infective phase represented by specialized objects or virions which serve as vehicles for introducing the viral genome into other cells. Viruses are thus parasites at the genetic level that regulate the synthesis of extracellular phase, the virion. You see the biological status of virus. Features of living organism in viruses: they are obligate parasites. They can multiple inside a living host cell. They contain either DNA or RNA as the genetic material. They exhibit variations due to mutations. They show irritability and respond to environmental conditions such as heat, UV rays, humidity, drought, alcohol, etc. We see the features of non-living objects. They have no cell structure that do not show cellular metabolism. They remain as insert particles outside the host. The viruses can be uh, precipitated just like chemical substance. Some virologists are of the view of that virus from a bridge between living and non-living things, that is, they are transitional forms between living and non-living. That is why viruses are placed under a separate group called a cellular microbes. To see general characters of viruses, viruses are submicroscopic parasite particles, a simple virus particle, often designated as virion, consists of a nucleic acid core enclosed within a protein coat called capsid. The capsid of some viruses is composed of a single type of protein. Various In some viruses, the capsid contains different proteins. The amount of protein in different viruses varies from 60 to 95%, and the rest is nucleic acid. You see the size of viruses. Viruses are of variable sizes. The size of virus vary from 20 nanometers to 20 nanometers to 300 nanometers in diameter. Poliovirus and viruses causing yellow fever and foot and mouth disease are very small viruses which are 10 nanometers in diameter. Pox viruses fall among the larger viruses which are rectangular in shape and measure approximately 400 into 240 into 20 nanometers in size. The dimensions of some common viruses are given, and if you see the parvovirus says 20 below nanometers, and turnip yellow mosaic virus 28 nanometers, and tomato bushy stunt virus is 30 nanometers, polio virus is 27 to 30 nanometers, adenovirus 60 to 90 nanometers, influenza virus 80 to 120 nanometers, and TMV virus is 17 into 300 nanometers. You see the shape of viruses. The viruses are of different shapes. They include rod-shaped, polyhedral, rectangular, and spherical, tadpole, and filamentous. Majority of the animal viruses are roughly spherical. You see the structure of virus, the complete assembly of the of infectious particles is known as virion. A virion consists of a nucleic acid core surrounded by a protein coat or capsid. This complete set of virion is known as nucleocapsid. The capsid is composed of a large number of subunits known as capsomeres. Chemically, the units of capsid are polypeptide molecules. Some viruses have an additional layer around the nucleocapsid called envelope. This envelope is made up of lipids, proteins, and glycoproteins. These are certain projections on the envelope known as spikes, which are arranged into distinct units. They help in attachment to host cell surface. If you see the nucleic acid, viruses contain either single or double stranded RNA or DNA molecules. The viruses containing DNA are called deoxyviruses. Various these having RNA are called riboviruses. Viruses vary considerably in the structure of nucleic acids. In general, all plant viruses have single stranded RNA, but cauliflower mosaic virus contain DNA instead of RNA. And animal viruses have single or double stranded DNA, but the polio virus which attacks children contains RNA. You see, bacteriophages contain mostly double stranded DNA. Filamentous phase contains single stranded DNA. Most of the insect virus contain ssRNA and only a few have DNA. And the nucleic acid may be in linear or circular form and have plus or minus polarity. The RNA within the virion is known as or sense strand because it acts as mRNA during replication. The RNA in some virions cannot directly act as mRNA. Instead, it acts as template to produce additional strand which may act as mRNA. Such RNA is known as antisense strand. You see the B is the protein coat. The nucleic acid coat of the virus is protected by a protein coat called capsid. The capsid consists of several identical protein subunits known as capsomeres. Each capsomere of TMV contains 158 amino acids. The bacteriophage contains smallest number of capsomeres in a capsid, but many viruses have as many as 162 capsomeres. These subunits are usually arranged in the helical or polyhedral geometric forms which are specific for a particular virus. The protein coat protects the viral genetic material and helps in its transfer between host cells. Yeah, and if you see the helical viruses, these viruses are shaped like a hollow cylinder. The nucleic acid strand is coiled like a helical spring. The protein subunits are helically arranged around the helical spring in a rotational axis. Then the helical capsids may be naked or enveloped. The polyhedral viruses. There are several animal, plant, and bacterial viruses which are polyhedral in shape and are either naked, poliovirus, adenovirus, or enveloped. Polyhedral structure has three possible symmetry such as tetrahedral, octohedral, and icosahedral. The viruses are more or less spherical in outline. Therefore, icosahedral symmetry is the best one of packaging and bonding of subunits, and icosahedron is a regular polyhedron with 20 triangular faces and 12 corners. The capsomeres of each face form equatorial triangle and 12 intersect points are kernels. Then if you complex viruses, you see the viruses which have the unique identifiable capsids are have the capsids with additional structures are called complex viruses. Example, Vaccinia virus and T-even bacteriophages. In these viruses, variations are also found. A definite capsid is absent. Example, Vaccinia virus. Capsid present and consist of a tail looking like a tadpole. Example, T-even phase. Capsid present and brick-shaped example pox virus and capsid bullet-shaped example nuclear polyhedrosis. Classification and nomenclature of virus is not authentic. You see the, for example, plant viruses. Now, the important plant virus is tobacco mosaic virus. More than 1,000 types of plant viruses are known which cause various diseases in plants. Of these, tobacco mosaic virus or Nicotiana virus has been studied most extensively. The virus was first discovered by Ivanovsky in 1892, but its isolation from infected plants and crystallization was done by W. M. Stanley in 1935. The chemical nature of TMV particles was revealed by Borden and Perea in later Frankel contract. Sanger and Williams confirmed that the genetic material of TMV is RNA. You see the structure, Franklin etc. all have described the structure of TMV particle. The particles appear as bundle of rods or needles under electron microscope. Each rod is approximately 3000 angstroms in length and 170 angstroms in diameter and has a molecular weight of 39 into 10 to the power 6 daltons. These particles have a protein coat and a nucleic acid core. The protein coat is made up of approximately 12 to 2130 identical protein subunits called capsomeres. Each capsomere consists of a single polypeptide polypeptide chain of 158 amino acids and its molecular weight is 80,000 daltons. The capsomeres are helically arranged around a central hole of 40 angstroms. Inside the protein coat, there is a single stranded RNA molecule which is also spirally coiled to form helix. with a pitch of 23 angstroms and at a radius of about 40 angstroms. The RNA does not occupy the whole but is deeply embedded in the protein subunits, thus having its own helix. The viral RNA consists of 6,500 nucleotides and has a molecular weight of 2.4 million daltons. To see the symptoms of our TMV on the host, the following symptoms appear in tobacco plant. Apical young leaves show downward curling and distortion. Unusually large motile light, green and yellow patches develop on the upper surface of the leaves. And epidermal cells and hair show lamella of amoeboid cells. If the plants are infected early in the season, they show stunted growth. There is distortion and discoloration of flowers. You see the viruses and plant diseases, symptoms of viral infections. The symptoms of viral infection can be grouped into following three categories. One is the external symptoms and second one is internal symptoms and third one is physiological symptoms. You see the external symptoms. These symptoms appear externally on viruses spots of the plant such as leaf, stem and fruit etc. First one is general symptoms. Some of the general symptoms common to most viral infections are general reduction in the growth of the host, reduction in the yield, and reduction in the lifespan of the infected plant. You see the external symptoms on leaves. The external symptoms on leaves may be following types: chlorosis and mosaic and necrosis and morphological changes and the external symptoms on stems. If you see, infected plants show many abnormalities. First one is external internodes become short and there is proliferation of lateral buds. It results in the formation of densely packed broom like structure. This symptom is known as witch's broom. Example, witch's broom disease of taprin. In some cases, the stem swells at certain points of human experience and due to excessive growth of tissues.